The mystery of Rh-negative blood. Could we be two subspecies?
Today we're exploring an intriguing hypothesis regarding the Rh-negative factor. Have you ever questioned whether all humans truly share the exact same biological lineage?
While modern science recognizes only one extant human species, a specific detail in our blood raises a fascinating question. What if we were actually divided into two distinct, silent subspecies?
What is the Rh factor?
It all began in 1937, when Karl Landsteiner and Alexander Wiener discovered the Rhesus factor.
At the time, it was believed that this antigenin human red blood cells was identical to the one found in Rhesus macaques, hence the name.
This system categorizes humanity into two primary groups: the Rh-positive lineages and the exceedingly rare Rh-negative lineages.
The primate enigma.
And here lies the first mystery. The International Union for Conservation of Nature currently recognizes over 710 taxa, including both primate species and subspecies. Curiously, however, not a single one shares the Rh-negative blood subtype.
If we are evolutionarily so closely related to other primates, why does this trait appear to be uniquely human? Where could this lineage have originated?
The Basque connection in Cro-Magnon.
The clue to this mystery may lie in northeastern Spain. The Basque people possess the highest concentration of Rh-negative blood in the world. They also speak a language isolate, one that does not belong to the Indo-European family, and carry genetic markers that predate the advent of agriculture. Some researchers speculate that this bloodline traces directly back to our Upper Paleolithic Cro-Magnon ancestors, representing a lineage that diverged distinctly from the rest of the global population.
Statistics and health, a taxonomic identity.
Today, Rh-negative blood remains a rarity. In Europe, it accounts for roughly 16% of the population. But globally, that figure drops to approximately 6 to 7%. We are talking about 500 million people who carry this biological marker.
But it's not just about blood groups. Research, such as the 2015 study by Jaroslav Flegr and his team, suggests that Rh-negative individuals may exhibit different incidences of certain health disorders and physiological states, reinforcing the idea of biological differences far deeper than we once imagined.
This is where the theory takes a dramatic turn.
A maxim in speculative genetics states, if we were to find two distinct genes operating with the same recessive antigenicity of the Rh factor in a single individual, science would no longer be looking at a simple blood variation, but rather a clear bifurcation in the evolutionary tree of the genus Homo. This would redefine Rh-negative from a mere blood type into a taxonomic species identity.
The 5 pillars of the great apes.
To understand human uniqueness, we must look to our closest relatives on the phylogenetic tree, the hominids, Hominidae. This family is characterized by a fascinating diversity of species and subspecies, which we can categorize into 5 primary pillars. The phylogeny of the family Hominidae reveals the complex evolution of the great apes. Branching early, the genus Pongo orangutans comprises the 3 species of orangutans, Sumatran, Tepanuli, and Bornean.
Moving to the subfamily Hominini, we find the tribe Gorillini, represented by the western and eastern gorillas and their respective subspecies.
The tribe Homi-H9a diverges into 2 subtribes, Panina, containing the bonobo,
and the 4 distinct subspecies of pan-troglodytes,
and Hominina, represented by Homo sapiens.
In this model, we highlight a critical evolutionary differentiation within the human lineage based on the Rhd factor.
This bifurcation marks the biological and reproductive implications of Rhd negativity, a central theme in our current theoretical research.
The hypothetical proposal, a biological minority?
In light of this evidence, its absence in other primates, its genetic uniqueness, and its distinct health implications, a provocative proposition arises: should humans of Rh-negative lineage be recognized as a distinct subspecies, or a specific minority group? If science confirms that this is indeed a separate evolutionary lineage, it may be time to consider legal frameworks and protections to ensure the care of this biological minority. This remains a hypothesis in progress, but one that compels us to look at our own blood through a different lens. What about you? What is your blood type? Let us know in the comments, and let's debate this idea.